Hey, good evening, everybody, and welcome to the Corner Society, probably the longest running history of medicine series in the United States. We've been around since 1935, almost continuously with a few pauses for world wars and pandemics, and excited to welcome you back. I'm Nicole Ross, and together with Bob Riley, we co-coordinate this series, and I'm having a really fun semester, and it's really extra kind of special to introduce a colleague and neighbor kind of conceptually from RIT. And so that local scholar that we don't get to hear a lot from. So this is Professor Christopher Whitney, who is an associate professor at the Department of Science, Technology, and Society at RIT. He received his PhD at the History and Sociology of Science program from the University of Pennsylvania. We met a speaker from that program who was a chair last month, and he completed a postdoc at the University of Wisconsin-Madison. His work on environmental science and policy garnered him the Best Article Prize from the Society for the Social Studies of Science, and he has received multiple competitive funding awards, including the National Endowment for the Humanities and the National Science Foundation. Anyone who's humanities knows kind of that's a big deal. He's presenting today from his really large-scale project, which is also going to be a book, about the many uses of horseshoe crabs. And I've had occasion to kind of look and read some of this material, and it's really a story about how crabs are at the basis of almost all we know and think about public health today, which, unless you're specifically immersed in this field, I think blows your mind. So kind of these crabs are small but mighty role in the ecosystem of public health in general. And I'm really excited to have the opportunity for all of us to learn more about crabs, public health, and medicine. So thank you for taking the ride down, and welcome to Corner Society. And join me in welcoming Christopher Whitney today.
Thank you, Mel, for the introduction. Thank you all for being here and on Zoom. Yeah, I'm going to try to convince you that horseshoe crabs are important, especially to medicine. And I should back up and say that this case study of the horseshoe crab is part of a larger project with some collaborators at Durham University in the UK on the use of marine biomaterials more generally. And this idea of, in the context of this idea of the blue economy, this idea that we can go to the ocean and we can extract resources, different materials, and use them for different purposes, cosmetics, nutrition, medicine, and so forth. But the question remains, what happens between that moment of medical discovery? Let's say you find a new compound in some sea creature in the middle of the ocean. What happens from that moment of discovery through to commercialization and incorporation of that material into biomedical practice and infrastructure? And the horseshoe crab is a very early example of this, and provides perhaps a cautionary tale for boosters of the blue economy idea. So if you've never seen one, let me see, try to advance this. There we go. These are horseshoe crabs, these helmet-shaped, spiky, ancient-looking creatures. There are 4 species of horseshoe crab in the world, 3 of which live exclusively in East Asia, and 1 of which lives in North America, only on the East Coast, from roughly the coast of Maine to the Yucatan Peninsula. They're ecologically quite important. They provide food for other marine life, like sea turtles, for example. They also provide habitat for other marine life. Horseshoe crabs are fairly long-lived, so they take 8 to 10 years just to reach sexual maturity, can easily live another decade beyond that. So once they stop molting, lots of stuff connects itself to them. So they are in and of themselves habitat for other creatures. They're also super important to shorebirds. What you're looking at here is a spawning event. They usually spend most of their time out on the continental shelf, but in May and June, mostly at the higher tides, they come in and they climb up on the beaches and they lay their eggs in the sand. This is timed perfectly, ideally, although that is changing, with the northern migration of numerous species of shorebirds who stop on the beaches and eat horseshoe crab eggs on their way to their Arctic nesting grounds. One of those shorebirds, the roof red knot, was listed as endangered in 2014, threatened rather, under the Endangered Species Act, in part because of a population decline in horseshoe crabs. So they're considered really important ecologically. They've been used by humans for a number of purposes, and I will go into a little more detail on each of these, but they've been used as fertilizer, they've been used as livestock feed, they've been killed outright as vermin for potentially predating shellfish beds. They're used as bait to catch other stuff, and they are used, which is why I'm here tonight, they are used in biomedicine extensively, because a component of their blood is used to test for endotoxins, pyrogenic substances in all intravenous drugs and medical devices. So if you have gotten a vaccine, and I assume we all have in the last few years, and you did not break out into a fever, potentially life-threatening fever, you can thank a horseshoe crab. So horseshoe crabs, I find, are representative of the technological and political complications that ensue when medicine and public health are predicated on the extraction of a natural resource. And horseshoe crabs have been considered as a model, sort of a model organism for decades. I have here just little snippets of testimony from the congressional record, different people talking about the importance of horseshoe crabs for very different reasons. The first one is Christine Stevens from the Society for Animal Protective Legislation, in her testimony to Congress in 1978 when they were considering amendments to the Endangered Species Act. She said, "Who would have dreamt that one of the most ancient invertebrates, Limulus polyphemus, the horseshoe crab, American horseshoe crab, would become an important animal model in the study of coagulation and hemostasis in man." A couple decades later, in 1992, John Burris, the director of the Marine Biological Laboratory in Woods Hole, is speaking to Congress about opportunities in marine aquaculture and biotech. And he's saying that one example of this use of new biotechnologies is the use of Limulus, the common horseshoe crab, whose blood contains a factor that allows us to detect endotoxins. And then just 2 years ago in 2020, Sudeep Parikh, from the American Association for the Advancement of Science, was testifying to Congress about the federal role in research and development. Again, using the example of endotoxins and Limulus amoebocyte lysate, LAL, as the gold standard for endotoxin detection, as a way to say it's really important that federal research dollars support basic research. So you have Limulus as this model organism, as this example used over and over again for the past 50 years about how important it is to conserve marine organisms, their potential for use in biotech, and how important it is for federal support of basic research. But our knowledge of horseshoe crabs goes back way further than that, in pre-industrial times, in fact. And these, I don't know if this is big enough for you, but this is a little game of find the horseshoe crab. Oh my, can I use this way? Yes, there he is. This is a 16th century English account, travel to Virginia. So European colonizers have known about horseshoe crabs from the beginning. This is a document, an image from a book describing some of the early uses of horseshoe crabs by the colonists and by the indigenous people there. Here he is again. This is a painting of a 17th century Dutch curiosity cabinet. So in the 17th century, horseshoe crabs start popping up in European curiosity cabinets as an example of an exotic species from somewhere far away. And this is not an image, but this is an excerpt from an 18th century English translation of Linnaeus. So by the 18th century, horseshoe crabs are being placed in taxonomies of all species by people like Linnaeus. Let me get my cursor back over here. Industrial times, horseshoe crabs are most prominently used for fertilizer. So this is an image from the 1930s along the shores of Delaware, and this is from the Delaware Public Archives. You can see the magnitude of this harvest. This is just piles upon piles of dead and rotting and desiccating horseshoe crabs. They would catch them in pound nets or pull them off the beaches, stack them up, wait for them to dry and desiccate, then grind them up and sell them as fertilizer, cancerine, crab-based fertilizer. So you see the magnitude of the harvest here. This is actually 50 years after its peak. This fertilizer industry probably peaked around 1880, when 4 million crabs were taken from the Delaware Bay alone. I should have mentioned back when I was discussing the ecology that the Delaware Bay, sort of a shallow estuary between the states of New Jersey and Delaware, is really the epicenter of this phenomenon. It's the largest spawning population of horseshoe crabs in the world, and therefore it's a major stopover for migratory shorebirds, and it was the epicenter of this fertilizer industry. So this is in the 1930s, and from the peak of 4 million crabs a year in the 1880s, it steadily declines through and dies out. This practice dies out in the 1960s, in part because of lower population numbers, but in part because in the post-war period, we had synthetic fertilizers and we didn't need to grind up dead crabs to put on farm fields. But also, as you can imagine, as the East Coast is developed, nobody wants to live next to this. You can visually see the extent of the fishery, but imagine the smell of this as all these crabs rot and desiccate. Nobody wanted to live near this. So it was in part pushback from Bayshore residents that this industry dies out. Horseshoe crabs are also well known to modern science and considered as model organisms in a number of different fields. This example is the work of Haldane and Keffer-Hartline was on the optic nerve of the horseshoe crab. Horseshoe crabs happen to have a very long and, as these things go, relatively simple optic nerve that people like Hartline use to try to understand how vision works, not just in horseshoe crabs, but in other species, including humans. And this work on visual receptors and retinal interaction, based on limulus optic nerves, won the 1967 Nobel Prize in Physiology or Medicine. Horseshoe crabs were also important in taxonomics, especially in this case, serological taxonomy. So for a brief time, relatively speaking, there was a serological museum at Rutgers University. And the person who ran that museum, Alan Boyden, his idea was we could compare species in a different way rather than just looking at, say, their body plan or different or embryology or all these other ways you can rearrange species in different categories. We can compare their blood, and by comparing species blood, we'll be able to sort of correctly identify them and taxonomize them. So this is also from 1967. This is an image from an article about the Serological Museum written by Alan Boyden's wife, Dr. Mabel Boyden. And she's talking all about how every May and June, they travel down from North New Jersey to Southern New Jersey, and they go to the beaches and they pick up all these horseshoe crabs. They bring them in, they bleed them, they separate out the different components of the blood, and then bring that blood back to the museum. And it was not only, and this is very typical, it's not only for their own work on serological taxonomics, but they become a supplier of horseshoe crabs and horseshoe crab blood to other researchers. So from here, from Rutgers, horseshoe crab blood is being sent out to other researchers in other places for various research programs. Probably the most well-known being a person at SUNY Buffalo, actually, who was doing a lot of work on cancer and using horseshoe crab blood for that research. Horseshoe crab blood specifically was also considered really important in terms of understanding, as a model organism for understanding how blood coagulates in horseshoe crabs specifically, but also in other animals as well as humans. So the top here is the title of an article, an 1885 article, Observations upon the Chemical Composition and Coagulation of the Blood of Limulus. by W.H. Howell. He was working at the Johns Hopkins Marine Station in Beaufort, North Carolina. And then this other title is from 1902, Leo Loeb. He was an MD in Chicago, but he was actually doing work on horseshoe crabs at the Marine Biological Laboratory in Woods Hole. And I mention these two specifically because they're important in the history of the study of horseshoe crab blood. They're cited by people for the next century, but also Johns Hopkins and Woods Hole specifically play a really outsized role in this history. LAL is invented, discovered at Woods Hole, which I will talk about shortly. And here it is. So the creation of Limulus amoebocyte lysate. The image here, the person on the left with the mustache is Jack Levin. The person on the right with the beard is Frederick Bang. It's Frederick Bang who sort of gets this ball rolling. In the 1950s, he's working with horseshoe crabs at the Marine Biological Laboratory in Woods Hole. And he's doing so not because he has some great insight into what horseshoe crab blood could do, but because they're there. If anybody's ever been to Woods Hole, it's on the coast of Massachusetts. It's well within horseshoe crab habitat. And so the laboratory would wild catch horseshoe crabs, bring them into the lab, keep them in sea tables and tanks, and they became a major supplier of horseshoe crabs to other institutes and academic settings for people doing research with them. So Frederick Bang these were just around. So out of convenience, and this is how he described it, out of convenience, he was using horseshoe crabs. He was like injecting bacteria into them just to see what would happen. And what he discovered when he did that was that some of his horseshoe crabs, their blood started to clot at the injection site. And so he looked into this for a couple of summers, ended up writing a paper in, I think, 1956 about what he discovered. And essentially what he worked out was that endotoxins, which are from the cell membranes of gram-negative bacteria, not gram-positive, only gram-negative, these ubiquitous bacteria, they're just everywhere. They have a toxin in their cell membranes that when it's released, horseshoe crab blood coagulates around it. So he worked out that it was only gram-negative bacteria, and he also worked out that it didn't have to be the bacteria themselves. He also lysed the toxin out of the cell membranes and exposed that to horseshoe crab blood, and the horseshoe crab blood coagulated around it in the presence of just the endotoxin. So and this is important medically because if there's endotoxin contamination in an intravenous drug or what have you, or a medical device, the bacteria don't have to be present. Like you can sterilize them with heat, but the endotoxin is heat stable and will still be there and will still cause a fever reaction to whatever it is. So it was important for him to figure out that it's not just about the bacteria themselves, it's about the toxin contained within the bacteria. And he published this paper and he sort of sat on it for about 10 years until he was joined by Jack Levin. So Bang was a Johns Hopkins medical researcher, a pathologist, an immunologist. So was Jack Levin. He joins Frederick Bang in the 1960s. And Levin really works out the details of how the mechanism by which this works. He discovers that the coagulant is not in the plasma of horseshoe crab blood, it's in the sole amoebocyte of horseshoe crab blood. So he figures out he can separate the serum from the amoebocyte, he can lyse the amoebocyte, and he can extract LAL. And then you can use just the LAL as an assay for the presence of endotoxins. He also discovers that the clotting cascade is actually a series of 3 coagulants. So this other picture, which is from one of his publications, is showing you the LAL reaction with endotoxin. On the left, clear, no endotoxin. On the right, in the presence of endotoxin, LAL becomes this milky clot. And you can actually turn the test tube upside down and it won't come out. It's still there. And it takes a matter of hours. Now, I should say, both Levin and Bang immediately recognized the potential, the medical potential of this product of LAL, because the prior way you test for endotoxins and other pyrogens, other fever-inducing substances, it was called the rabbit test, the rabbit pyrogen test, where you inject whatever it is you're interested in into the ear of a rabbit, and you monitor the rabbit for 2 or 3 days to see if it breaks out in a fever. If it does, you have a problem with your batch of whatever it is, vaccine, whatever. If it doesn't, you're okay. That's an in vivo test. You can imagine the expense and complication of maintaining colonies of rabbits just to inject them with stuff. This is an in vitro test. It's much faster. It's also much more sensitive to the presence of endotoxin. So they immediately saw that there was a lot of potential for this medically. By the way, we still do use rabbits for endotoxin testing because it turns out that endotoxins are not the only pyrogen. And so even though LAL has largely replaced the rabbit test, we still maintain some colonies of rabbits. Nobody knows where they are or how many there are or how often we use them. But for some purposes, for some pyrogenic testing purposes, we still use rabbits. But we use way less of them, would be what a proponent of LAL would say, is that we went, in animal welfare terms, we went from using tons of rabbits all the time for this to just using a few in select cases. So Levin and Bang are thinking about this mostly clinically. They're thinking, oh, maybe we could test for sepsis in the human body using LAL and different sort of clinical applications. But a different person altogether sees the larger commercial potential of LAL. James Sullivan and Stanley Watson, also working at Woods Hole, although not at the Marine Biological Laboratory, but rather at the Woods Hole Oceanographic Institute. Stanley Watson, he's using LAL, or trying to use LAL to test for bacterial biomass in seawater. Just a sort of ecological study, has nothing to do with health or medicine. But he can't get enough of the LAL, so he starts making it himself. And he actually, in 1977, it was filed in 77, he actually was awarded the patent in 78. He patents a method for creating LAL in a more efficient and sensitive way. And he goes to the Woods Hole Oceanographic Institute and the Marine Biological Laboratory and says, hey, do you want in on this? Could we produce this together commercially? And this is sort of way before universities and academic institutions think about basic research as a way to make money, right? So they're like, no, we're not interested. Why would we do that? So Watson, who had started a real estate company with his wife called Associates of Cape Cod, repurposes that pre-existing organization as a company that will now make LAL. He gets approval from the FDA to do this also in 1978, 77, 78. And he starts making LAL in his basement and in his garage, and then eventually rents a larger facility to produce this.
Synthetically,
Bleed horseshoe. Oh yeah, I should explain that.
Yeah.
You need live wild. Thank you for asking that. You need live wild horseshoe crabs for this. So into this, I forget like basic information. You can't grow horseshoe crabs. I mentioned they're really long time to maturation. You can't just put them in a tank and wait around for 8 years for them to spawn so that you can have more horseshoe crabs. You have to collect them wild, and you bring them into the lab and you take roughly 1/3 of their blood by poking a needle into the membrane around the heart, and you bleed them. That's the blood you use for making LAL, and then the horseshoe crab you put back in the water. The modern LAL industry will tell you that the mortality rate from doing this is in the low 3 or 4%. 3 or 4% of crabs undergo this procedure and die. Some academic biologists will tell you, no, the real number is more like 30%. The people in charge of regulating this fishery, which I will discuss in a moment, they use the 15% number, sort of in the middle. So they estimate that of all the crabs brought in from the ocean or the beaches, bled in a lab for making LAL and put back in the ocean, about 15% are going to die, which doesn't account for any sublethal effects. That's a whole other thing. So Watson is harvesting horseshoe crabs or paying fishers to harvest horseshoe crabs for him. brings them in, creates LAL, and is selling it. And the FDA, he gets FDA approval for this in 1978. Within a year, there's 4 other companies / people doing the same thing. And I'm thinking this is a super obscure process, and perhaps it is, but I included this really weird comic strip because this is a 1978 Buzz Sawyer comic describing this process of how horseshoe crabs, quote unquote, donate their blood for the benefit of humankind. And so this is a really interesting example to me anyway of this being in the air in the 70s, that people other than researchers that were told know about this somehow. See what the.
Do you want to read it?
Would you like me to read it?
No, I can't quite see it.
Oh, sure. In the first, see if I can see it. In the first panel, the scientist is saying, first we insert the needle into the dorsal area, and the man is saying, poor beastie. The person's name gets his first lesson in bleeding the crabs. He asked, will it kill them? The woman says, quite to the contrary, they simply donate a little of their blood to save human lives. And then the man says, like going to a blood bank. Exactly. Then we tag them and return them to the sea. So it's being portrayed rhetorically as this benign process to save human lives right from the very beginning. And this is actually resonates very strongly with the way that the contemporary LAL industry talks about the process. The language of donating, blood donation, is ubiquitous on their websites and their literature. Now, the path to full FDA acceptance was actually pretty long, about 15 years. These are just excerpts. The top is an excerpt from the Federal Register when the FDA is first announcing that they will be looking into using LAL as a replacement for the rabbit test. But until such time as they approve it, you have to keep using the rabbit test. And what a lot of pharmaceutical companies were doing was running both in tandem as a way to generate data on the comparability of LAL and the rabbit test. So a lot of people were doing both, which is what the market for LAL was at that time. It was pharmaceutical companies looking to use LAL as basically a QA/QC tool, a way to test the water they were using, a way to test the components of their drugs, under the assumption that if their components went into the process pyrogen-free, the product would be pyrogen-free too. But by December 1987, the FDA issues their guidelines, and now it's okay to just use LAL. You don't have to, in most instances, you don't have to use the rabbit test anymore. So in that 15 years, pyrogen testing goes from injecting substances into the ears of rabbits to bleeding horseshoe crabs in the lab and using LAL for in vitro testing. Now, as LAL producers are really quick to point out to this day, they have always been about conserving the horseshoe crabs. They would say it's in their own economic interest to conserve horseshoe crabs. If there are no more crabs, no more way to produce LAL. So in 78, as the FDA is first starting to take a look at the production of LAL, they're also drafting the quote unquote return to sea policy. The idea that horseshoe crabs, and this is from the Federal Register as well, horseshoe crabs shall be returned alive to their natural environment after a single collection of blood and shall be handled in a manner so as to minimize injury to each crab. And this again is, LAL industry would say, this has always been our policy. This is still our best management practices to this day. We treat the, I have a fisher who, LAL companies don't go out and grab the crabs themselves. Of course, they contract with fishers who go out and trawl or collect crabs for them. And I have a fisher sort of on record saying, we treat these crabs like babies. We treat these crabs better than my granddaughter. Okay. So they're very adamant. They treat the crabs well. They put them right back where they found them within 72 hours. And again, even though the official mortality estimate is 15%, possibly 30, they say no, it's more like 3. So they're very in on the idea that the LAL industry is actually a force for conservation. But because this is a wild caught animal, marine invertebrate, the FDA is not the only regulatory body overseeing all of this. The other body is the Atlantic States Marine Fisheries Commission, the ASMFC. Because in the 1980s and 90s, a new fishery emerges. Horseshoe crabs start being pulled out of the water in huge numbers and collected off the beaches during spawning events in huge numbers to serve as bait. So fishers would collect the crabs, chop them into pieces, and use them in pots to catch eel and conch, whelk really, but referred to as conch. Most of which were for foreign markets. So they catch these animals and then ship them off to the Mediterranean and East Asia. This is a chart from the ASMFC showing the harvest from the late 90s to 2020, and those orange lines are the commercial bait harvest. So you can see in the late 90s, the numbers of crabs being harvested are approaching those historic numbers. Well over 2.5 million crabs pulled from the water just to be chopped up and used to catch other stuff. This was of concern to biologists, conservationists. It was also of concern to the LAL industry because a lot of these people remembered or knew of the fishery, the fertilizer fishery from 50 years before, and knew that that fishery had really decimated the population. And because they take so long to reach sexual maturity, it took a really long time for their population to bounce back. So everybody except the bait fishers are calling for regulation of this fishery. It had never really been regulated before. It was tracked before in the days of fertilizer, but it was never regulated. Nobody ever told fishers that they couldn't take a certain number of crabs. And in fact, as I mentioned, they were actively killed as vermin in some contexts for fouling fishing nets or eating shellfish. So the ASMFC installs its first fisheries management plan for horseshoe crabs in 1998. There have been 8 addenda since. The most recent one was adopted 1 week ago, and each addendum has slowly reduced the harvest quota for horseshoe crabs. Similar to the fertilizer fishery, the ASMFC tracks the biomedical harvest. That's the yellow lines, and you can see now more crabs are pulled out of the water to produce LAL than are pulled out of the water to serve as bait. And then they use that 15% mortality rate to establish those little green lines, the number of crabs killed by the biomedical harvest. And then they add that up and incorporate into their harvest quotas. So these mortality numbers are included in the harvest quota, but the ASMFC does not and maybe cannot directly tell the LAL industry how many crabs they may or may not take. In fact, the number of crabs taken by any given facility or company, there are 6 of them now, is proprietary. We don't know how many each company takes. We know coast-wide how many crabs are harvested, or we think we know, but we don't know, like we can't break it down by locality. So they don't tell the biomedical industry that you can only take X number of crabs. They just sort of track it and incorporate the death of those crabs into their harvest quotas for the bait fishery. So it's a really odd situation. The LAL is in a really odd situation right, in terms of regulation. They're overseen by the FDA, they're monitored by the ASMFC, and they bristle at the idea that they would be regulated by either. As it happens, what am I doing on time here? Okay. As it happens, there are alternatives to LAL, and there have been for 2 decades. One of them is called recombinant factor C, or RFC. This is a recombinant product produced with recombinant DNA technology. You take yeast, you splice in a gene that tells the yeast to produce this factor C, which is the first step in the coagulation of horseshoe crab blood. And this has been around, like I said, for about 2 decades, commercially available since, I'm going to say 2005 from Lonza. But it's not widely used, and it's not widely used because the FDA has not given its full approval to use RFC instead of LAL, despite a lot of pressure from environmental activists and certain parts of the pharmaceutical industry to do so. And this person standing here heroically on the beach is Jay Bolden. He works for Eli Lilly. He's a big birder, has a real passion for protecting shorebirds by protecting horseshoe crabs. And he has led the charge for the last decade at Eli Lilly. Eli Lilly is probably not the first, but the most significant adopter of RFC. They decided 10 years ago that all their new products would be tested with RFC instead of LAL, which the FDA allows as long as you validate that test every single time for every single product and prove or show that it's equivalent to using LAL. And Lilly can afford to do that, right? They can afford the different equipment they need. They can afford to go through the validation process. But it's been much slower to catch on in other sectors of big pharma and little pharma and medical device companies because FDA hasn't given its approval, and the FDA hasn't given its approval because the US Pharmacopoeia hasn't given its approval. And so there's all sorts of shadowy conspiracy theories about what's happening at the US Pharmacopoeia that they haven't approved this yet. So it's become quite a heated controversy. I have a student working with me now, and I've shared a bunch of my interviews with these folks, as well as some of the historical documentation. And she was really amazed at how rancorous, how grumpy everybody is about this, how finger pointy they are. Environmental advocates blame the FDA and the USP. The USP says we don't have enough data. The industry says, the LAL industry says RFC isn't the equivalent. So it's become quite a heated controversy. There's another alternative called MAT, and this is actually a human blood-based test. So this would involve actual blood donation by humans to use components of human blood to test for pyrogens, including endotoxins. This is also a working test. It's accepted in the European Pharmacopoeia, though not in the US one yet, as of yet. The benefit of this test is that it doesn't just test for endotoxins, it tests for all pyrogens, because it's looking for what happens when fever-inducing substance is in blood. So from animal welfare advocates, this is the test to go to, because it not only replaces horseshoe crabs in our public health infrastructure, it replaces rabbits completely. And the person I talked to at PETA thinks that the future is some combination of MAT and RFC. So MAT in instances where you have to test for all pyrogens, RFC for more QA/QC in the pharmaceutical manufacture process. So here I'm sort of gesturing at that political controversy I mentioned. On the left is an image, a screenshot from Charles River Laboratories. They are the largest LAL producer. They're also a supplier of laboratory animals generally for all manner of medical testing and pharmaceutical testing. They have a whole website dedicated to horseshoe crab conservation. On the right is the logo of the Horseshoe Crab Recovery Coalition. This is a loose network of environmental NGOs, really prominently Audubon because of the shorebird connection, but a lot of other animal welfare and conservation organizations. And what's interesting to me about these two opposing groups of people is they're very different uses of the past. They're very different framings of the past. To the LAL industry, they've always been conservation minded, and their practice imbues former vermin with value. They make horseshoe crabs more valuable because the horseshoe crab is the source of this substance that is one of the cornerstones of modern medicine, modern pharmaceuticals. To the environmental activists, this is anathema. That they point to demonstrable effects of overharvested horseshoe crabs on horseshoe crabs themselves, but on all the other species that depend on them. And they consider it a completely unnecessary extraction of natural resources when there are viable, in their opinion, viable alternatives. Okay, I am going to finish up on time here. Back to the beginning. Frederick Bang again. In 1981, less than 2 weeks from his sudden, surprising, and untimely death in 1981, he gave a lecture to a group of endotoxin researchers, again at Woods Hole, titled Rules and Regulations Regarding Serendipity. And he tried to talk about the horseshoe crab instance as indicative of science more generally, and that it often involves a lot of serendipity. Happy accident awaiting the prepared mind. So, and this is how he describes his discovery of LAL in the 50s. He happened to be using horseshoe crabs, but he was ready to see what happened to the horseshoe crabs if he put different bacterial toxins in them, because he was an expert in febrile reactions in the human body, and he had been talking to colleagues about the Schwartzman reaction, which is a similar reaction that occurs in rabbits. And because Woods Hole was this convivial place where people were always talking about the research, all these things combined to instantiate this happy accident, the serendipitous discovery. But that's just the discovery, right? What Bang could not have imagined was the tremendous industrial and bureaucratic inertia that would have to be overcome to switch from the rabbit test to the LAL test, and now in the attempt to switch from the LAL test to RFC and MAT. He also, I don't think, could have quite envisioned the moral and ethical implications for public safety, ecosystems, and animal welfare that this really complicated system became. What a historian of technology would call a large technological system, right? This huge network of technologies and peoples and policies that make LAL part of our public health infrastructure, and also make it really difficult to change that infrastructure. So back to sort of my initial question, what happens when medical discoveries in non-human nature are scaled up to become medical products? Proponents of the blue economy might keep in mind that this process is so much more than discovery. Like finding a chemical in some plant or animal in the ocean is just the beginning. And it may make public health dependent on ecosystems in really complex and ethically impactful ways. And I will stop there. Thank you.
All right, thank you so much. That was awesome. I'm going to use this for questions because I think there are people on the Zoom so that they can follow along.
How did you become interested in this issues?
Serendipity. I used to live on the Delaware Bay Shore. And when I went to grad school, which happened to be in Philadelphia, I wanted to pick a topic of research that I cared about. And so I cared about the bay and its ecological denizens. I told Corinna right behind you where she could go to see this massive spawning event one year. And so for my initial work on this system, I initially focused on the shorebird connection, the shorebird researchers. But then again, serendipity, and actually this is a great excuse to switch to my acknowledgements slide. A colleague, Dr. Elizabeth Johnson, right there, emailed me one day a couple years ago and said, do you know anybody who knows anything about horseshoe crabs? I was like, I know something about horseshoe crabs. Here's an article in a journal I just published on the fishery. And she was putting together this team funded by the Lever Home Trust to look, this is the larger project I mentioned about marine biomaterials, to look at different case studies in the use of marine biomaterials in health and medicine. So I said I would look into the horseshoe crab, biomedical horseshoe crab fishery. And we have an artist, Dr. Helen J. Bullard, and a postdoc, Dr. Hannah Dickinson. They're looking into jellyfish, the biomedical uses of jellyfish, and the use of shrimp shells for chitosan, and the use of other shellfish for chitosan. Yeah, so that's how it started. And then I got was lucky enough to get some NSF funding to really devote some time to really researching this. I've collected hundreds of documents, dozens of interviews, and hope to spend the next year, as I'm happen to be on sabbatical, working this all into a book. So I'm sort of in that fulcrum where I stop collecting things and start thinking about things and writing things. So I appreciate the opportunity to think out loud and give you sort of an overview of the project.
I think you said that they tag the crabs after they take the blood. So does that mean they don't use them again? Or so they won't harvest them again?
So, so you can't, there are several tagging studies. Some of them are run by Fish and Wildlife Services at the state and federal level. What that comic was gesturing to was actually what the industry does. And I can give you the example of Lonza. They're located in Maryland, but their LAL facilities in Maryland. Lonza is obviously a big international pharmaceutical company. What they do is they contract to trawl their horseshoe crabs. And after they bleed them, they put a notch in the shell on the right or the left side. And that tells the fishers next year not to bring those horseshoe crabs in, to put them back in the water, because they won't bleed them 2 years in a row.
Okay, but they do regenerate their blood, so
Yes.
they could be rebled later on.
They could. Lonza wants to make sure it's been at least 2 years. But they could be bled again. And in fact, I didn't mention this. There's an attempt to have a horseshoe crab ranch by a small biotech firm in South Carolina. And their idea is that if you keep these horseshoe crabs in tanks and keep them nice and healthy and feed them good food, they'll be healthier. The LAL produced from their blood will be better, and you can bleed them more frequently. That's very much in the sort of biotech phase. He offered to let me come down and see the place, but then he turned his laptop and showed me that it's a room about this big with like 2 big tanks, and that maybe it wasn't worth my time to go down there. It's very much unclear whether that will work out. But yes, in theory, you could bleed them again. There's also concern about sublethal effects, though. Like, does a crab going back into the water with a third less blood mean that that's a crab who's not going to spawn that year, or is going to be confused and not make it back out to the continental shelf or what have you?
Do we still don't know answers to that, like so many years into this?
We don't. And in the history of biology, it's surprising how little we know about any animal, but the horseshoe crab, marine animals are particularly hard to understand because you can't just sort of walk around behind them. They're really hard to track. They molt so much in those first 8 years, you can't really put a tracking device on them because when they molt, it's not on them anymore. So they're really hard to track. They're really hard to find. They're really hard to understand. They spend most of their lives completely out of sight. So yeah, we don't actually know a lot about them.
But you will learn.
But people are trying to.
Seems like bleeding them more often is going backwards.
Yes, but what the tech bro who runs this place told me is that this is a chance to stop using wild caught horseshoe crabs. That if you can maintain a steady population in captivity, you could, in theory, bleed them more often and use that LAL. There's also been advances in how we use LAL. So there are different technologies, cartridges that use less LAL per test. So if you could use less LAL per test and use LAL from captive horseshoe crabs, theoretically, maybe, if you could scale it up that far, you could stop using wild caught horseshoe crabs. It's very much theoretical at this point, though.
It would be like using the rabbits that you have your own horseshoe.
Yeah. Instead of having rabbit colonies, you'd have horseshoe crab colonies. Vast tanks of horseshoe crabs.
Fascinating. I'm wondering if you could, I'm going to ask you to speculate about the future of the blue economy.
Mhm.
And it just seemed like you're really at the end of this talk describing an inflection point where we've gone from a state of discovery and moved to a point very quickly with the monocyte activation test and the recombinant factor C, that there is no longer a need to exploit that environment other than for discovery. And as we move to that sort of an economy where the blue wealth of the world becomes just a place for discovery because we can do the rest recombinantly, how do we protect that resource?
That's a great question. So because of some of the work that these other folks on this team are doing, I can tell you that horseshoe crabs really are an early example of exploiting. It wouldn't have been called the blue economy in the 1950s, 60s, and 70s, but the same idea, exploiting, extracting resources from the ocean to use in health and medicine and for other purposes. Horseshoe crabs is really an early example of that, and the potential model or cautionary tale, depending on your perspective. And a lot of the other imagined uses of marine biomaterials are very futuristic. Like maybe we could do this, and maybe we could do this. Maybe we could grow shrimp on Mars and use their shells for chitosan. And chitosan could be used for all these other things like sutures and for nutritive purposes. So it's very much in the like futuring model. And I suppose, to be speculative, I suppose if you really could be assured that anything you discovered in the ocean could be created synthetically using recombinant DNA, you could limit your resource extraction in the ocean to just discovery and then make everything else with the yeast or bacteria in vats. It's not clear to me. And if that's the case, then yes, the horseshoe crab example is a good model of how you could cut out this whole period of intensive resource extraction and just go straight to the recombinant product. Because one of the reasons I think everybody would agree that we haven't just switched to RFC and MAT is because the LAL industry doesn't want to. They're making a lot of money doing this. And they consider themselves a force for good, et cetera. But if you can cut out all of those several decades of industrial extraction and just go straight to recombinant DNA technology, sure, maybe the blue economy could just stick to discovering and make good on those promises. It's not clear though. It's really unclear whether all this futuring could actually be produced, made real. And it's also unclear if everything that we're talking about taking out of the ocean could be produced synthetically using recombinant DNA technology. And I think the chitosan.
What is the chitosan?
So that's the material. It's actually in horseshoe crabs too. It's the part of the material that the shells are made out of. So invertebrate shell, horseshoe crabs, shrimp, other shellfish, lobsters, I assume, invertebrate shells have this material that has been known that we've been using for like medical sutures for a really long time and grafts and things. But it has all these other imagined uses as well that I'm not completely familiar with. But there are certain material, the point being some materials we're imagining taking from the ocean and using on an industrial scale could potentially be produced recombinantly, but others perhaps not. So it's still worth looking at the devil's in the details, right? It's still looking at the specifics of each context. Why are you researching this particular organism? What do you imagine its uses to be? And if you can imagine and make real a use on an industrial scale that would benefit a lot of people, what are going to be the consequences of that use, intended or otherwise?
So what are the main factors between the regulatory disparity in the US and in Europe? You mentioned that Europe has adopted some of these more synthetic processes and the US hasn't. Is that mainly just the power of the industry here, or are there other factors?
There are, I think, two major factors. You're right. European Pharmacopoeia has accepted not only MAT and RFC, but is like really dedicated to getting rabbits and other vertebrates out of medical testing. And And most people don't realize this, but the US actually has a similar law enacted in, I want to say 2000, that created, I'm going to remember the acronym, but not what it stands for. It's called ICFAM. And it's basically a cross-agency committee across federal agencies, USDA, EPA, FDA, all these different USGS, all these different agencies. And they have the mandate to look for, wherever possible, how to use fewer animals in cosmetic testing, medicine testing, pharmaceuticals, all this stuff. But it doesn't have quite the same teeth as its analog in the EU. And the EU, it's much more of a mandate. They can step in and say, you can't use rabbits anymore for this purpose. In the US, it's more like a committee that gets together and studies the potential for using fewer rabbits, for example, but can't really mandate that FDA, okay, now you must do this. So I think most people are surprised to learn that we have such a law, but even though we have it, it doesn't have quite the same teeth as its EU analog, as I said. The other major factor here is exactly what you said. And if you're more cynical or maybe even conspiracy minded, and on the environmental activist side, you're saying this is all about industry. And I've had people tell me in interviews, the moment that Charles River has their own synthetic version of LAL, this will all go away. The RFC will be approved and we'll stop using LAL. But until that time, they're going to fight tooth and nail to keep producing LAL. And in fact, Lonza produces both. They were the first company to hold the patent and commercially produce RFC, but also they produce LAL. They produce and sell both. ACC, the one started in Woods Hole, the first LAL producer, they have their own recombinant product now that is supposed to be even better because it doesn't just mimic one coagulating factor, it mimics all three factors in the cascade. So in theory, even the LAL industry, with the exception of Charles River, is on board with this change. But Charles River is the holdout, because Charles River, they're working on theirs and it isn't ready yet. Or at least that's the cynical take, which probably has more than a hint of truth to it.
The price of the difference, like how much would it cost to do a final of each, and how much is it sold for?
I wish I knew that. I do not know that. I wish I knew that. That's something to look up. You've given me something to look up. Thank you.
What's revenue more money?
Yeah, it may be. I don't see any questions in the chat.
Wait, is that the chat or just?
This is the chat and this is the participants. So I don't see any questions yet.
I wanted to ask a question from, oh, you got it now?
So I have the history of technology question.
Uh-huh.
I want to see the scale of this industry. It seems like such a pre-industrial process, right? Like hauling them in, tagging everyone, every single one individually, eating, and there's so much labor and time involved in this process, when usually when we think about history of technology, mass production is all like interchangeable parts, you know what it is.
Yeah.
So I want to see the scale of this, and I know I read an op-ed in the New York Times 10 days ago, I think, in anticipation of the recent regulation, and the anticipation was that these numbers would go up, that the harvest quotas would go up, and you were just telling us that they actually seems to have gone down. So I'm pleased for the conservation effort here. So that's my history of technology question. If I can tack on another one, like based on the dialogue that we had about the blue marine economy, I am wondering, there seem to be different goals that you are talking about. You are talking about the conservation of the blue economy, actually turning it into a non-economy almost, saving it from exploitation, whereas the project that you are part of, it almost seems like an exploration to like open up the blue economy. So I was wondering if you could like talk a little bit more about the bigger picture ideas there regarding the blue economy, and what this is this an idea about conservation, about exploitation to conserve other parts of the natural environment? Like, what is behind it really?
Okay, I'll start with the last one first. So this team is STS people and cultural geographers. And so there's actually an inherent skepticism of the claims of the blue economy because it is so future oriented. It's a lot of almost boosterish rhetoric about the potential uses of the ocean and marine biomaterials. And so I think most of the team actually feels the need to like interrogate that. Like what is the purpose and ramifications of this rhetoric? Is this actually being done? Or is it still very much sort of the imagining phase? So yeah, I would say that there's sort of a interested skepticism on the part of the team toward this blue economy idea. In terms of the scale, so this gives you a sense for the scale in terms of the harvest. We're still talking about 500,000 animals dredged up and bled per year for the LAL industry. It is labor intensive. Fishers don't make a lot of money. They're not paid a lot to do this, but they're paid enough to do it. So as labor goes, they're treated as relatively low labor workers. I can tell you that the labs themselves end up hiring a lot of seasonal workers because this is not an all-year thing. This is when the horseshoe crabs are available for harvest thing. So they can hire seasonally, bring a bunch of college kids into these labs to do the actual poking and prodding and bleeding, and then take it from there. In fact, I have talked to a student on the RIT campus who said she did this for a summer in Massachusetts. She works in one of these labs. And then when she told me who her boss was, I said, oh, he won't talk to me. So I probably shouldn't ask you any questions either. But it was nice of her to tell me that she had done this. So that's the scale in terms of harvest. I would say as a business, LAL industry is something in between tech startup and big pharma. Like it's a very small part of Lonza's so even though this is a ubiquitous substance and used ubiquitously as quality control in pharmaceuticals worldwide, so it's big on that scale, but as a business, producing a product, making a certain amount of money, we're not talking big pharma numbers here. This is a very small part of Lonza's business. This is a very small part of Charles River's business.
By the way, I just looked it up. Save on Aldrich, you can buy each of these steps for about $1.50.
Oh, really? Okay.
Well, both, I guess. Can you tell me, just a small question first, why did the yellow only start in 2004 when obviously they've been harvesting biomedical collection prior?
As I mentioned, the AFMFC monitors and encourages the industry to adopt best practices and takes the death of the mortality of some of their crabs into account, but they don't actually tell the industry what to do. And so they didn't even bother monitoring until 2004. So there was a change in the fisheries management plan around that time that was made so that they would start paying attention to the biomedical harvest as well.
So we don't know the scale, but we can assume it was similar preceding 2004.
Yes. Yeah.
So my question, this is really interesting about technology, about the blue economy and stuff, but I wonder if I could kind of encourage you to tell us or think, what can we learn for public health? What are the public health lessons to learn about a public health intervention that is actually so rooted or so dependent on this animal derived factor? So she's asking her a technology question, I'm asking a public health question, we all ask, where we're coming from. But
right. Well, so one line of thought, I can give you what other people have had to say about this. One line of thought is that the industry is overstating the case when they say that this is a life-saving product. That actually endotoxin contamination is relatively rare, and that it's rare to actually cause fevers. And if it does cause a fever, it's very, very rare to cause death. So one lesson would be, beware inflated rhetoric from pharmaceutical industries. I think that applies to a lot of things, not just LAL. Another line of thought is, and this is what conservationists today say, is that if we don't have to tether our public health system to the extraction of a natural resource, especially a wild animal whose populations can fluctuate for all manner of reasons, having nothing to do with biomedical interest. Why would we tether our public health system, the purity of our public health, of our pharmaceuticals, to such a thing, to such a natural cycle, in some ways out of our control, when we don't have to? And there again, I think that's for people imagining the uses of non-human nature, bits and pieces of animals and plants, and chemicals derived from the ocean or elsewhere in medicine. We should think about, will we have to be picking up crabs and jabbing them with needles, or whatever the analogous situation would be in another sector? Or are there better ways to think about what we choose to tether our ability to keep pharmaceuticals safe, effective, endotoxin, or whatever toxin free?
All right, well, thank you so much. That was a wonderful talk, an interesting topic. Thank you. I'm sure everyone joins me in thanking Professor Christopher Whitney. I want to invite you to come back for the last talk we have for the semester. It's December 7th. We have Dr. David Cantor from Buenos Aires coming to talk about cancer research and educational film in the mid-20th century. He has a book that came out with University of Rochester Press. I'm sure there'll be a lot of clips to be seen. And so that will be the last one for our semester. Then we have an exciting spring program. And let's all talk about crabs a little bit more as we have the food right over there. Thank you.
Thank you.